Okay, well, good evening, everybody. It's great to see so many people out tonight, so thank you very much for coming along. My name's Prof Helgen. I'm the Executive Dean of the Faculty of Sciences at the University of Adelaide, and it's my pleasure tonight to introduce Professor Kris Helgen, who's giving his inaugural lecture, and you can see the title, The Life of Mammals in a Fast-Changing World. Firstly, I'd like to acknowledge the Kaurna people, the original custodians of the Adelaide Plains, on which the University of Adelaide's campuses at North Terrace, at Waite and at Roseworthy are built. I could give you a whole lecture on Kris's record, but I'll have to give you the potted version because you want to hear him, not me. Kris's research focuses especially on fieldwork and research in museum collections to document the richness of life, to explore global change and to contribute to important problems in biomedicine. He's published hundreds of scientific articles, and he regularly publishes in Science and Nature, the two leading journals. He received his undergraduate degree in biology from Harvard University and his PhD in zoology here at the University of Adelaide. From 2008 to 2017, he was curator in charge of mammals at the Smithsonian's National Museum of Natural History in Washington, DC. And he was overseeing the world's largest museum collection of mammals and the most visited public gallery of mammals in the world. He's worked professionally in more than 50 countries, and he's documented and named dozens of previously overlooked species of living mammals, including the skywalker hoolock gibbon of southern China, the greater monkey-faced bat of the Solomon Islands. I'm a botanist, this is really hard work. And the Olinguito of the Andes Mountains. His expeditions and discoveries have been featured in two major documentary series by the BBC Natural History Unit. He holds professorial or other honorary academic appointments at George Mason University in Virginia, the American Museum of Natural History in New York, the Bishop Museum in Hawaii, the Australian Museum in Sydney, the South Australian Museum here in Adelaide, the National Museums of Kenya in Nairobi, and the National Geographic Society in Washington, DC, where he was inducted as a National Geographic Explorer in 2009. There's many more things I could say about Kris, but it's an absolute pleasure for me to say that he's now a staff member at the University of Adelaide, and I'd like to hand over to Kris so he can give us his inaugural lecture. Thank you.
Most importantly, you can hear me? Great. We know it's a professorial lecture because, like all good lecture hall lectures, we have people clustering in the back and students in the back here. Thank you, Bob, for the kind introduction, and it's good to be back here in Adelaide. Thank you. I want to say thanks up front to all the different collaborators and institutions and funding bodies and students in particular who have helped to produce the research that I'm going to talk about today. I want to echo my thanks to the Kaurna people on whose land we come together today. Let's get started. How rich and how varied is life on Earth? With how many species, past and present, have we shared this planet as our home? How are species distributed across the globe? Which ones are common, which are rare, which are endangered species? How do they come together to work and form ecosystems? What do they do for a living? These are some of the absolutely most fundamental questions that occupy the minds of scientists in universities and natural history museums who study biodiversity, the richness of life on our planet. And these are the kinds of questions that have occupied my mind since my earliest days. And I do mean my earliest days. This is me. I'm 3 years old, and although my command of fashion may have declined, my interests haven't changed really very much. And you can see, my mom sent me this picture. I was already at this age fascinated with animals, and I was fascinated kind of sorting them out. The horses, go with the horses. The story in our family is, this is me getting a Noah's Ark toy set. And if you remember how the story goes, the animals come 2 by 2. So the horse goes with the horse, but then there's something about them that puts them close to the zebras too, right? And so that was the kind of question that flew into my mind. I loved animals, cats and dogs and horses, but to me, there was a more fundamental question in my head, which was, how many kinds of animals are there? How many are there? And when I posed that question, increasingly, that was always asked in terms of mammals. Mammals were what interested me most. And so this is me at 3 years old. I grew up in Minnesota, 8 years old, at the Crooked Lake Public Library in Coon Rapids, Minnesota. I found a book called The Encyclopedia of Mammals. It's actually my own copy from back in the day, 8 years old. And I thought, finally, well, here's where I can find the answer to that question. How many kinds are there? Here's a fundamental reference. It was sort of somewhat popular, somewhat scientific, and a thick tome. And so I started taking it home and studying it. And eventually, I wore the copy so ragged that the librarians took me aside and they said, look, you've worn out this copy. We're going to buy another one, but now it's going to go in the reference section, and you can't take it home. And my parents took pity on me. They bought me this copy. But the truth is, I studied this volume. As a little kid, 8, 9, 10 years old, I knew the scientific name of essentially every mammal on our planet. And I learned the terminology of mammalogy. And the different species involved, like a child, would essentially learn an additional language. That's something that very rapidly, as I grew up and started working with mammalogists, including some people like Tim Flannery in my teen years, that brought me rapidly face to face with some of the most remarkable animals all over our planet, like this animal, the largest egg-laying mammal, the long-beaked echidna in New Guinea, and bringing me close and working hand in hand with some of the most remarkable people on our planet, like these two gentlemen who live in different parts of Papua New Guinea, who I soon learned could teach me so much more about the animals that live in their landscapes and their lands than I was ever going to learn from books. And this has involved expeditions, as you heard from Bob, all over the world, many different countries, hugging the equator, the tropics of the world, Africa, Southeast Asia, South America. Here's a picture from New Guinea. This is a helicopter landing down in the Foja Mountains of the western part of New Guinea. This is an area so remote, hundreds of kilometers across, rising to 3,000 meters nearly up into the sky, that as far as we know, it's never been inhabited by people. This section of the island is part of the mountains. So there's no tracks or roads, there's no villages, there's no hunting, there's no dogs. And imagine what it's like. You can set up a makeshift camp there and be the first people to come in and look and see what kinds of animals are there. This is the mission. Let's go to these places and see what's there. And I should say, this question when I was a kid, asking, how many kinds of mammals are there? Where do they live? This, as I grew older, allowed me to start making maps and models of where I thought the gaps are. Where are the blank spaces on the map where we don't really know what's happening? These are the places where I've been going for almost 20 years. Imagine the excitement. Here's putting a live trap, we call an Elliott trap, on the ground here in these Foja Mountains. The excitement of putting this trap on the ground in the afternoon and the evening, coming back, you're going to come back in the morning and look inside, and you might have the chance to look in that trap and see a kind of animal, a kind of mammal that maybe not only no Westerner has ever seen, no scientist, perhaps no person has ever seen before. That's the thrill. That's the excitement of, getting into this kind of work. And it can lead to amazing discoveries and rewards. This is some of the species that I've been able to name. This is an animal called the blue-eyed spotted Khuskhus. This is a marsupial. It's found only on an oceanic island, never connected to another landmass off the coast of New Guinea. Beautiful. This is one of my favorites. This is something called the greater monkey-faced bat. This is found only in the Solomon Archipelago. And this bat has a larger set of jaws and teeth than any other bat on the planet. So in some measures, this is the largest bat on the planet. How is it that the largest bat on the planet, arguably, it doesn't get its scientific name until the 21st century? Well, it helps you start to understand how little it is we really actually know about our natural world, especially in places across the tropics. This is a bat that in the grand rainforests of the Solomon Islands, where no other terrestrial mammals have really arrived, there's no primates, there's no other kinds of mammals that we find in most other forests, these bats have evolved to eat the thick-skinned fruits and the heavy nuts that other mammals would eat elsewhere. So this is a bat doing something that we don't see elsewhere. Another of my favorites is a meter-long giant rat that we found on one expedition to an extinct volcano in the south-central part of New Guinea, Mount Besavi. This rises like a volcanic cone out of a flat tabletop landscape. And as far as we know, this rat, which was tame, we could approach this and handle it without fear. This rat is found only, as we so far know, inside the caldera of this extinct volcano. And it's not just rats and bats, it's extraordinary animals, some quite big. This is an ape. This is a gibbon, a smaller species of ape from the Asian mainland. It was mentioned in the introduction, the Skywalker Hulak gibbon. This is a species named earlier this calendar year, 2017. And this is something that we've known as scientists for 100 or so years, lives in a little pocket of southern China, now a protected area. Very endangered species, but no one had looked close enough to see exactly what kind of gibbon it is. So the name is based on a classical Chinese allusion, the notion of, these animals feature in ancient Chinese literature, their graceful motions through the canopy are kind of likened to heavenly movements. And so Skywalker is that translation, but some of our team, especially from China, were big fans of Star Wars. And Mark Hamill, who is the actor, Luke Skywalker himself, got really excited about this and started tweeting about it. This discovery reached hundreds of millions of people. People love these kinds of things, and that's an opportunity for me and other scientists who do this kind of work to engage broadly with the public. And it's also not just using techniques that we've always used, but delving into new kinds of techniques, like camera traps. This is just as simple as putting new kinds of camera technology into the forest and seeing what's living there in a way that you would never be able to experience just by going there and sitting in the forest and watching and waiting. This animal, one of my favorites. This is a tree kangaroo from New Guinea. This is called Dendrolagus pulcherimus. Pulcherimus means most beautiful, and this is called the most beautiful tree kangaroo. This animal, this particular kind of tree kangaroo, was known only from one mountaintop in Papua New Guinea, discovered in the mid-1990s. We thought there were about 20 of them anywhere in the world. Going to the Foja Mountains, that isolated place in western New Guinea, we rediscovered a new population of these animals that's quite safe across the expanse of this mountain range. So, but yeah, this is an animal taking its own picture with a camera set up on a log crossing a gully. Probably has crossed this gully, many or most nights of its life. And all of a sudden here we have a picture, the camera is flashing. It's taking a little closer look. You can see the beautiful markings, and backs up and decides to try another way across. Gorgeous species. Or this animal, the tiger. We were using camera traps just a few years ago in a section of northern Burma, Myanmar, where tigers were thought to have become extinct. It was using these cameras and placing them all through these forests that allowed us to see that there was a second chance. We hadn't lost this animal yet in this part of this country. And this is, again, some of the rewards of this kind of work. So this kind of work brought me, at a very early age, squarely into the realm of a place where you might not expect, which is natural history museums. Because this is the kind of place where these fundamental questions are usually addressed. How rich is life on Earth? And when we, whether members of the public or scientists, think about natural history museums, we're often, mind's eye is thinking of these public galleries where we could see dinosaurs or mammoths or elephants. Or maybe the old-fashioned kind of view of specimen cabinets that are, open to the public and people can come through and browse about natural history. But when I think about museums, in my mind's eye, this is what I see. Row after row of these kinds of, white metal cabinets. And this is where the magic happens. These are the treasure troves behind the scenes that the public's generally not ever going to see, where treasures, specimens from expeditions past, decades, even centuries ago, have come and amassed and are stored inside these cabinets. And this is where we go to try to figure out these thorny problems about how to tell species apart and how to plot their distributions. And you can find treasures in here. Here's one of my favorite specimens. This is a long-beaked echidna that I found in the Natural History Museum in London. Now, it was not enjoying its best life as a museum specimen. It was buried in the bottom drawer within the museum of where long-beaked echidnas are stored. And it was buried under sort of echidnas 3 deep. And it was in there because it was thought to have no known locality. It was a useful specimen, but we don't quite know where it came from, so perhaps not as valuable as some. But when I saw this specimen, and by the way, I was interested in this question of these big echidnas known only from New Guinea. How many kinds are there? Across the different rainforests and mountain ranges of New Guinea, is there just one species? Are there several? Which ones are endangered? These questions. So I wanted to see every echidna specimen, long-beaked echidna, in every museum in the world. That's actually what I did. So whether this had a locality or not, I wanted to dig through and find it and look at it. But when I did, it stopped me in my tracks. And that's because I recognized the tag. I knew this tag, which was personally printed for the collector John Tunney. Now, John Tunney was a pioneering exploring through the top end of Australia, but he never went to New Guinea. And all of a sudden, I was surprised. This animal's only known from New Guinea. And this is Tunney's tag in his handwriting, which others had missed. Now, the locality here says Mount Anderson in the West Kimberley. I thought, what could it be? Could it be that this specimen, which was collected, we know the date, in November 1901, was a 20th century record of the long-beaked echidna in New Guinea. We knew that this is Tunney on his pioneering adventures up in the Kimberley. We knew the long-beaked echidnas used to be in this part of Australia. Why? Because they're in rock art. We have fossils from the Pleistocene, the Ice Ages, and there's some notion of perhaps an aboriginal knowledge of them in the past. But did they get not through to the Ice Ages, but all the way through to the 20th century? Maybe they're still there today. So these things can go hiding, and they pose interesting questions. Can we go up to the Kimberley today and find a long-beaked echidna? Another specimen. This is a story that takes me to the Field Museum in Chicago, another of the great museums of the world. And imagine again those rows of white cabinets. I opened the drawers on one, and I pulled out this long, big and heavy metal drawer, and in it I saw these fluffy kind of orange-red skins. Now, it's not exactly what I expected to see. Like with the echidnas, this is a group of mammals I'd been studying across all the museums of the world. This is a member of the raccoon family, a group of animals called olingos and kinkajous, Latin American tropical rainforest raccoons living up in the trees. But this was like nothing I had ever seen before. Nothing I'd ever seen had these pelts, and there were in the same drawer boxes that had skulls of these animals. These had been in this museum for about 60 years from an old expedition. And I looked at the skulls, and instantly I thought, this is not like anything I'd ever seen. Could it be that this is a specimen that all zoologists have overlooked, a kind of species of mammal that all zoologists have overlooked? And so if it was there 60 years ago, I wanted to know, is it still there today? We used the information from the tags on these specimens, which showed that they had come from parts of the Andes of Colombia and Ecuador. We decided, well, where would be best to go and look, picked a place, knowing that these kinds of missions can be like a needle in a haystack sort of thing. Went down to some of the glorious cloud forests on the western slopes of the Ecuadorian Andes. And the first night that we were there in the forest, we found this animal, which is called the Olinguito now. This is a species that we introduced to the public as a new species, named for the first time in 2013. This is in a group of mammals, the carnivora, which includes the dogs and cats and bears and raccoons. No one had named something new in this group in the Americas, North or South America, for almost 40 years. This is a group where we thought this kind of work is largely done. So again, it just shows us by looking a little bit closer, taking in every specimen, looking a little differently. That's how science works. Sometimes it throws up things that are quite unexpected. Now, the Olinguito is found secluded safely in these beautiful cloud forests up in the Andes. It lives in its own little private world. But increasingly, our world looks more like this, right? It's a human world dominated by our species, humanity. And it's an extraordinary thing. All kind of amazing aspects of the planet that we can start to think about, whether it's our ability to look up and see the night sky, the chemistry of rain, the temperature of our planet, all of these kinds of things. We're living in an era where they can actually be affected by us and our impacts on the planet. It's extraordinary. And it's so extraordinary that scientists have seen fit to maybe label this new era in our time here on Earth as the Anthropocene, the age of people, so changed by human impact that we have to see fit to give it a new geological term, a new era. And this has got me thinking, as I've traveled the world and seen these kinds of extraordinary environmental impacts wherever I go, it's got me thinking more and more the kind of work, this detective work in museums that I'm doing. What can I be doing more in this era of the Anthropocene? How should we be proceeding with these kinds of studies, understanding mammals, the life of mammals in this fast-changing world? So it got me thinking about museums. Now here's where you earn your chops at the lecture, right? Pay attention. A little potted history of science in natural history museums, right? So 1758. If you remember your biology from high school, you know that Carl Linnaeus in Sweden, comes at the thorny problem in the mid-1700s of how do we figure out how many different kinds of plants and animals there are on the planet. This is clearly important economically, for agriculture, for anything we can start to really think hard about, and we want to know how rich is life on Earth. And Linnaeus is audacious enough to imagine he was going to solve this question and this problem within his own lifetime. Still nowhere near the solution we know now. But 1758, part of Linnaeus' system in trying to understand this, not just dividing things up into genus and species that we still use today, but had to do with objective evidence, understanding that if we're going to start to tell species apart, we need to compare them closely, different leaves on plants, different skulls on mice, and we need places like museums to do it, where we're going to tuck away the hard evidence, the bodies of these plants and animals, so that scientists can come back and back and back again to verify the differences between them. That's how we're going to figure this problem out. So it's not necessarily the most intuitive thing, right, that we should, for time, immemorial, tuck dead plants and animals away into cabinets, so that we can come back and back to them. But that defines a lot of the science of biodiversity. So Linnaeus was interested in the units of life, right? How many species there are, the richness of life. 100 years later, Wallace and Darwin come to the most fundamental conclusion in the field of biology, this understanding that life is changing all the time and that it's evolving through natural selection, through means of inheritance. And this fundamental insight allows them to start thinking about reconstructing the history of life. If life is sharing common ancestry, we want to know something not just about the units of life, but its history, its relationships of life. And 100 years on from there, in the 1950s, the first time scientists start to understand that it's DNA that is the molecule of heredity. But even with that fundamental switch to understanding what you can use genetics to study these problems, we're still basically in what I'm calling Acts 1 and 2. Acts 1 documenting, like Linnaeus wanted, the diversity of life. And Act 2, like Darwin got us started on, is reconstructing its history. These are the kinds of, if you're a modern biologist, working on modern specimens of plants or animals, these are the kinds of studies that we're mostly involved in natural history museums. So let's step back. I was interested in thinking, well, is there an Act 3? These things could hardly be more important, right? Understanding the richness and history of life. But is there an Act 3? And I think there is. And I think it is basically surrounds this question of how do we document human impacts and the way that the world is changing. And these same specimens that I'm showing you coming out of these cabinets in the natural history museums of the world, they're recording things collected 50, 100, 200 years ago. And those same specimens that we use to tally up the units of life and its relationships, we can also, because it offers us these sort of time slice, these sort of time capsules from different times and places of animals and plants, we can reconstruct these things. So what am I talking about? We often talk about museums as vouchering life, meaning kind of giving a hard basis, a hard evidence, backing up what we know about life on Earth. And that has to do with, again, the units of life, how rich life is. It has to do ecomorphology, anatomies, comparative anatomy, comparing these things. It has to do with understanding distributions, genetic diversity. We're interested not just in understanding the relationships of how things are related genealogically or genetically. We're also, of course, as scientists, interested in how things come together ecologically and form ecosystems. For example, what animals eat which plants, or which pathogen is associated with which flea, which lives on which rat, in what part of the world. These are fundamental but really, really difficult problems. And these are the kinds of problems we solve with Linnaeus's system of tucking away specimens into cabinets and coming back to them. But in Act 3, we're interested in not just vouchering life, but vouchering global change, the way that life is changing very fast. And it just so happens that these museums are probably our best tool for getting at this change. So not only are we interested in a catalog of these measures, but we're interested in sort of a calculus of them, right? So how is diversity changing? How are anatomies transforming? How are the distributions shifting or shrinking or growing? How are relationships changing as the world changes? These are what we might be after. So quickly, a specimen like that Olinguito, or the long-beaked echidna, tucked away in a drawer, has a few simple sources of information, but they are very, very powerful. So for every specimen in a museum, we tend to have an identification, some notion of what it is, and a locality, some idea of where it was collected. And as simple as those two things are, when you sum them up across the diversity of life, those two simple pieces of information have basically bequeathed to us the entire sciences of systematics or classification and biogeography, or the distribution of life. If we take all of those specimens in total, that's what makes those scientific disciplines come to life. And traditionally, that identification comes out of studying a phenotype, an anatomy, a morphology. We look at a hippo and know what it is, but increasingly that's done through genetics as well. We can rely on DNA to help us hone our identifications of specimens in museums or in the world. But these are the kinds of things that have been really important in those Acts 1 and 2, the diversity, the relationships of life. But in Act 3, we can take advantage of the fact that most museum specimens also have a timestamp. We know when they were collected, usually by whom, and so we can use that to understand change through time. And here I've added genetics and other biochemistry, because as these sciences have matured, here in the 21st century, we can increasingly take advantage of the very simple fact that it's the bodies of these plants and animals tucked away in these drawers. These bodies preserve the chemistry, for the largest part, of what they were like in life, still in death. And so what am I talking about? Nucleic acids, proteins, stable isotopes, radionuclides, even contaminants and pollutants. These bodies tucked away in drawers can really help us to reconstruct all kinds of environmental aspects. And I've put up there other dimensions as well, because there's always been other sources of information for specimens like this. It might be photographs of them, the field site where they were collected, or something like that. And as we think about Act 3, the changing globe, photographs from decades or centuries ago become ever more important. So we're interested in all kinds of information that help us reconstruct these things. So now we find ourselves in Act 3. And the way I think about this for museums is that museums, for the rest of science, have long been this really important, strong pillar, where museums serve this role of vouchering these aspects of life. But now I think in the 21st century, to really derive their greatest value and to defend their value to broader society, we need to think about this second pillar, where museums have these two incredible strengths at least, studying diversity and history of life, but understanding its change. I'll give you a few examples from my research program where we can think about this second aspect of global change biology. One aspect that helps us start here is a pretty simple thing, extinction, right? Some species that used to be present on our planet are now gone. And for the most part, if we want to study them empirically, get our hands on them and understand something about their biology, we have to go to museums to access those specimens. Up in the corner, we see the passenger pigeon. It's a North American bird. This was one of the most abundant vertebrates in the entirety of the North American continent until about the early 1900s. So found mostly east of the Mississippi, it's now extinct, and it's been extinct for more than 100 years. The last one died in a zoo in 1914. Those kinds of things start to get us thinking about, how can we really understand the natural ecological context of North America if what's arguably its most abundant vertebrate has been extinct for 100 years. We can think of lots of Australian examples too. My favorite is the thylacine. What an extraordinary animal. Found not just in Tasmania historically, but prehistorically all through the entirety of the Australian mainland and up into New Guinea. And this, of course, was the dominant large predator of the native mammal fauna here in Australia. How can we really understand how Australian ecosystems work, how they're supposed to tick, when this animal is extinct and has been for tens or even thousands of years in different places? So extinction gets us thinking about major change, but there are all other kinds. Here's an example of extinction. Not long after I finished my PhD here at the University of Adelaide, I went back to the US, and I had never yet been, at that point, to the Academy of Natural Sciences in Philadelphia, which is our oldest natural history museum in the US. So being so old and just a tremendous collection, I knew that when I finally got there, there would be treasures that I could really uncover. And I had studied the genus Pteropus, the flying foxes, very intently as a PhD student. So I went to the Academy of Natural Sciences, and I started, the first time I went there, got in the door, went into the drawers, and started with the species of flying foxes. And within 1 hour, I had found this, which is an unnamed species of flying fox, the first hour that I was in the museum. And this was so exciting. Now, this was a small skulled species that had good information about being collected in Samoa, one island in the Samoan archipelago in 1856. This species has never been seen since. A US Navy mission collected this single specimen. Its skin is in a jar of alcohol. And its skull I had to piece together, did different fragments, and put the teeth back in to figure out what it was. Now there's two species of flying fox that live in the Samoan archipelago today. They're both much bigger than this species, and they're both something of ecological generalists. They both eat sort of medium-sized fruit. This species was doing nothing like that. This was probably eating little flowers, little inflorescences, or tiny fruits. This would have been playing a very different role in the pollination and seed dispersal of Samoan environments. Having found it, and giving this window into what was going on in Samoa in the 1850s, I wanted to see, are there more? Can I find other specimens of this species in other museums? So again, I did my museum trip to find out, are there more? And I didn't find any more of this species, but I found 2 specimens in the Smithsonian of a species that was found in Samoa, collected in 1838, but was much larger than either of the 2 species living there today. So the collections from the 1830s to 1850s that the first scientists who arrived in Samoa made show us that there were 4 different kinds of flying foxes that were dividing up the ecosystem there in Samoa. The smallest and the largest became extinct in the 19th century. The 2 more generalist still survive. I think this is giving us a window into what's going on all over the world, which is that many extinctions have gone unseen. And it's only through very careful detective work, getting into some of these older collections, that we make the fine comparisons that allow us to understand not just that these extinctions occurred, but that our understanding needs to change, that these ecosystems, how can we really understand them if there are only half as many flying foxes doing many different things, or doing much different roles in the environment than there were in the past. This is one of the beautiful flying foxes that's there today. But these are strong interactors in these insular ecosystems. There's no other mammals that arrived out on these islands naturally, so these are the mammals that are playing most important roles in the lives of the forest, the pollination, the seed dispersal, et cetera. Another is the concept of resurveys, right? So there are extraordinary past expeditions where people went from museums out to all parts of the world, major collections, maybe sometimes staying months or even years in one place and sending them back home to London or Belgium or wherever. But there's probably about 100 to 200 of these really core, important collections that allow us to really understand by studying them what the ecosystems in those places were like at some very high level of complexity. One of my favorites is Teddy Roosevelt, the US president, just had finished his tenure in the White House. This is 1909. He finishes in the White House 3 weeks later. It's an era of shipping. He's already on the coast of East Africa, and he mounts the largest safari field expedition of all time as the outgoing U.S. president. He's there in Africa for about a year in different parts of Kenya. And one of the best and most amazing collections was made on Mount Kenya, which is the second highest point on the African continent. Now, Teddy Roosevelt and his naturalist compatriots made very fine collections all the way from the base of the mountain to the tops of the glaciers. And that data today, we can use that to go back to this mountain, which we've now have done, and to reconstruct those kinds of studies in exactly the same places to understand this glacier has almost shrunk to nothing. The vegetation zone seems to have changed. How has the animal life changed in tandem? And it's a complicated mountain that with many different floristic zones. But of course, we can use these new techniques now that I'm talking about. We don't just have to go like Roosevelt did and shoot these animals. We can use camera traps to record the lives of aardvarks or bushbuck or leopards on this mountain and compare that with data from the past. And here's a quick, hot off the press discovery from our ongoing work on Mount Kenya. This is an animal called a potto. Who's heard of a potto before? Okay, Bill. This is a primate. This particular kind of potto is found only on Mount Kenya. This is a specimen of it in the Nairobi Museum. This gives you an idea of what it looks like in life. No photograph of this species because it hasn't been seen since 1938. 1938, no one has documented this species living in Mount Kenya. Now, this is a project that's being led by Marianne Guattari, who's a postgraduate student at the University of Nairobi and whose studies are tied and funded by our projects in Kenya. But Marianne went out, you saw a picture of a leopard. She wanted to know, what are leopards eating on this mountain, and how might that have changed through time? And so she collected as many scats as she could over months of effort all over this mountain. And she genotyped them. She used genetics to confirm that they were leopards, and then she used genetics to understand which different kinds of antelopes and other animals are in these scats. But 2 of the scats that she thought might be leopards turned out to be hyenas. It's kind of unusual. Hyenas live up in these mountain forests in a very different kind of African context than we see them elsewhere in the savannas of Africa. But these 2 scats that were of hyenas, genetically, both of them tested positive for having this animal, the Mount Kenya potto, as part of the prey. And looking a little bit closer, she was able to find little bits of the body parts and microscopically compare the hair and confirm that this indeed was potto coming out of here. So this animal, despite being, this is one of the most visited national parks in East Africa by hikers and mountain climbers. This animal hasn't been seen since 1938. Marianne and our team has just rediscovered this based on these approaches using scat and genetic techniques and cameras to reconstruct the hard-to-see lives of these animals. That's pretty exciting. Climate change. That's another thing that we're interested to know a lot more about, and these approaches of studying the past museum specimens can help us reconstruct things. This is a book that I recently published at the Smithsonian, which was based on incredible archival accounts, writings from the 1880s by a man named Lucien Turner. He was hired as a meteorologist studying weather from Smithsonian to go and be stationed for 3 years up in the eastern Canadian Arctic and to record sort of minute observations about ice and climate and temperature. But alongside that, he was an incredible kind of polymath. He mastered the Inuit language, the Eskimo language. He mastered the language of the inland Native Americans. And he studied the birds and the mammals and the plants and everything that lived in this place while he was basically isolated from the rest of the world for 3 years up there. Thousands of pages on the life of mammals are in the Smithsonian's archives that Lucien Turner developed through his interviews with Eskimos, his own hunts, and the information from fur traders in the region coming and going. One of the more interesting things, and by the way, these document, through to today, remarkable changes in temperature, changes in ice, changes in the populations of the caribou, the extinction of the wolverine. One of my things that I took most notice of is that Lucien Turner reports 3 kinds of bears: the black bear in the forests, the polar bear, of course, out on the sea ice, but also something he called the barren ground bear, which he thought might be the brown bear of North America. And what we're looking at, where Turner was working, was way up here. This is the modern, this is the fossil, recent and current distribution of the brown bear or grizzly bear in North America today. Nowhere do we really know of brown bears occurring to the east of the Mississippi. But here's Lucien Turner from the 1880s in these unpublished archives arguing that there are brown bears coming from fur traders in this part of the world. Now, he wasn't able to pay for them. They were too expensive, but he did collect Native American amulets that hunters would use when they'd go into the territory of the barren ground bear to protect themselves from what they considered this very ferocious animal. Now, these turn out, these amulets, to offer us the only hard evidence that's still around that this bear did exist. But if we go back into other information, including recent archaeological digs that have pulled bear skulls up out of Native American middens, and other kinds of historical observations, we realized that the grizzly bear was indeed there, like Turner said, and that it probably actually persisted in the most remote parts of this area called Ngava until the Second World War. So the grizzly bear, one of North America's largest terrestrial vertebrates, supposedly living nowhere east of the Mississippi, persists all the way through the 1800s, probably even to the 1940s in this section of Canada. And these examples, whether this is a story kind of like the long-beaked echidna and the Kimberley, they remind us of how little we actually really know about the distribution of life, the different kinds of species and where they live, especially because these things have changed. These things have changed in recent generations and even within the span of our own lives. So we're talking about these two pillars for museum science, and the more the technology changes and science develops, the more we can delve into these kinds of problems and learn extraordinary things. The University of Adelaide has a tremendous facility and group of people that study ancient DNA coming out of these kinds of museum samples, or even going back thousands of years from fossils and the like. We can study what are called metagenomes or microbiomes, understand the microorganisms that lived on different species going back in time. This is exciting. This is a disease called white nose syndrome. Some of you may have heard about this. This was something I was studying back in the US. This disease arrived in the northeastern US in 2006, apparently from Europe. And it started spreading through populations of hibernating bats. It presented as a fungal growth on the face, and it started killing our bats in the United States. In some places, killing up to 95%, tens of millions of these bats. And it has spread through more than half of the United States and many different Canadian provinces. So it's been there 10 years, and it's wiped out a huge percentage of the bats in this part of North America. Where does it come from? I thought, well, like all these other questions, let's go into the museum cabinets and see what we can learn. We screened hundreds of bats from different parts of North America, from different parts of Europe and Asia, and detected the first occurrence of this fungus from 1919 in a cave in France. This sits well with what epidemiologists have suggested about where this disease might have come from, but there it is, sitting in a cabinet, a specimen with hard evidence. We can go back to this very specimen, detect this fungus, and know that that fungus lived there and then. This is extraordinary, and this is something that's come out just this week in a journal called Emerging Infectious Diseases. I've produced a series of papers recently on koalas. It's a wonderful animal. This is our koala, but specifically about Koala Retrovirus, a really interesting virus. So just over a decade ago, the human genome was sequenced for the first time. And genomics has transformed since that time. But one of the most important insights when these genomes, the full length of DNA from us and other species, started becoming available was the insight that something like 10%, maybe between 7 and 12% of the genome, all the DNA of every mammal species looked at, comes from viruses. There's about 10% of our genome that's not really human. It's viral in origin. It comes from viruses that have infected us at some point in the past, and they have integrated themselves into becoming part of our own genome, part of what makes us human. or another kind of mammal. And some of these viral origins in the genome actually turn out to be involved in very, very important things that make mammals mammals, including placentation and the way that we reproduce and have young. But anyways, coming back to the koalas are the only wild mammal that we know of that has, at the moment, a virus that both can spread from individual koala to koala, like a normal disease, but also from koala parents to offspring by integrating into the sperm or egg. And this is the only single virus that we know that can transfer in both of these different ways. Could it be that Koala Retrovirus gives us the best place where we can look as biologists to understand how a virus integrates itself into becoming part of the genome? Well, this work has been funded by the U.S. National Institutes of Health. It's been considered so important that a group that funds fundamental work in cancer and virology has seen fit to support this work on koalas. And some of the work that I'm doing right at the moment has to do, too, with Tasmanian devils. Everyone has heard of the devil facial tumor disease, which is remarkably a contagious cancer, right? A cancer that can spread between individuals by transfer of cell lines. I've been using specimens tucked away in museums to go into past decades and understand how the DNA of devils has changed in fundamental ways that might help us understand the genomic basis, the actual basis at the level of DNA into how this contagious cancer was born. Do we want to know more about how a contagious cancer comes to be? Yes, we do. So if this kind of work from historical samples is important for reconstructing the richness of life and the way the world is changing, we might even think of a third pillar where we can explore important questions in biomedicine through these specimens that have usually been used to study biodiversity. And these are big, big questions. How does a virus become part of us? How is a contagious cancer born? How are the most important and virulent infectious diseases transferred between different species or parts of the globe? It turns out that these kinds of samples may actually offer us the best tools we might have, and that scientists who study biodiversity may be poised to, just as much as a physician, a biomedical researcher, to ask and address and perhaps answer some of these questions. Now, technology is changing. We talked about biochemistry. We can think about digitization, the way that specimens can be visualized and images can be swapped all over the world. And this kind of work that I'm talking about is going to require various kinds of theoretical and quantitative advances, statistical approaches to help to reconstruct the past from specimens that weren't really collected for the purpose of these kinds of studies, but nevertheless are very useful for them. And this is going to also involve new kinds of training. The way that we've trained biologists in different parts of biology, whether it's to be a biomedical researcher or to be someone who studies plants or animals, that's going to have to change, and it needs to be changing right now so that we can be thinking in these new ways and solving these problems together. So I'm going to close. I was in the Botanic Gardens just a couple of weeks ago, and this is a place close to my heart. This is where Lauren and I were married here in Adelaide back in 2006. But I came across this Museum of Economic Botany, and it had been some years since I'd seen it. I went in, and it's been remarkably refurbished. I think many of you may have seen that, you know that, and I thought it was fantastic inside. And it got me thinking, this idea, this Museum of Economic Botany, this is something that's been here in Adelaide since the mid to late 19th century, and there's almost kind of a quaintness about it, economic botany. But like these kinds of things I've been talking about, really little could be more important. These are fundamental things. You go inside and you understand how scientists in a colonial era were trying to understand how plants from this new part of the world or brought to this new part of the world, how they would flourish or not, and how they could impact the local economy and society. And cities, big cities all over the world, had these kinds of institutions. Now today, in the 21st century, we don't really call it economic botany. That sounds quaint because it kind of is, but we call it other things. We call it food and agriculture. We call it wine. We call it pharmaceuticals and medicine. We call it soil and water. And it's these kinds of institutions where we derive the first and most basic understanding of all of these kinds of things. You know, it's expensive to build a museum of economic botany or a natural history museum in the world's great cities. And there are good reasons, good and practical reasons that governments have always seen fit to do so and to maintain them. And part of it has to do with studying the richness of life, but other parts of it have to do with studying very practical and profound questions that impact economies, impact societies. And I've given you some examples of those today. But if you go back into past writing, into the 19th century, through the 20th century, these are the kinds of questions and problems that people in universities and people using natural history museums are thinking about, are researching, and increasingly with new technologies, these are things where we can make major impacts on fundamental practical things, commerce, agriculture, wildlife and medicine, et cetera, using these kinds of materials. So I'm back. I'm glad to be back. I'm very, very excited. These kinds of questions and problems that I've been talking about, these constitute some of the real grand challenges in academics, in science, that the university here in Adelaide is very interested in pursuing. And these are the kinds of questions that I intend to pursue, that I intend to pursue with leadership here at the University of Adelaide. I intend to do that in partnership with some of these, drawing on these venerable partnerships like exist between the university and places like the South Australian Museum. And I hope to be doing this and pursuing these studies in collaboration with other University of Adelaide faculty, and especially with University of Adelaide students. So it's nice to be home, and thank you.
We do have time for a few questions, and there are a couple of microphones doing the rounds, so if you want to ask a question, just put your hand up and we'll get you a microphone so everyone can hear it.
Hello, I'd like to ask, did he find out about the Tasmanian Devils?
Where are you? Where are we, over here? Great. Yeah, Tasmanian Devils, incredible animals. What did we find out? It's work that's kind of still in progress, but what we've found is that it's similar to the study of koalas, looking at koalas or devils. Some aspects of the genetics of these animals are changing in small ways through decades and centuries when we make these comparisons, but some things are changing in larger ways. And one of the things I will say is, we looked at white nose syndrome in bats. We can study the microorganisms that live on the skin and get into the bodies of these animals. And what we think is happening with devils is that over time, a particular kind of bacterium that lives on the body of devils, and devils are scavengers, they have lots of bacteria all over them. We think that particular kind of bacterium that wasn't in Australia a long time ago but came to Australia with European impacts might be involved genomically, it might be involved in the origin of this disease. So stay tuned on that. But you can see, I think, from examples like that, the power of these kinds of approaches. It's a lucky thing, again, that these specimens, whether it's skins or bones, have been tucked away like time capsules for our study.
One over here, yep.
Your trip to New Guinea, is it Fogia Mountains?
Yeah.
Are you thinking of going back there, or is there an even better place you could go?
Yeah, so great question. I mean, that was a life-changing experience to get into a place like that. Like I showed you with Mount Basavi and the big rat, we found in the volcano. Similarly, in the Fogia Mountains, animals there are less wary of people, a little more tame than you see anywhere else in New Guinea or the Australian kind of region. So I went there once in 2005 while I was still doing my PhD here, and I went back in 2008. Across the last 15 years, I've had the chance to do similar studies in many of the least explored isolated mountain ranges in New Guinea. So the answer is we do want to go back to the Fogia Mountains. I think there's a lot more still to be discovered there. We never used camera trap techniques there in the forests. So I'd like to get back there, maybe leave cameras even up for a few months, leave them there and come back and see what we can find on those. So that's exciting, but we're also still planning to mount expeditions to even even places that we know even less well now than the Foges.
Okay, we've got one just over here.
What was the largest species of mammal you've discovered?
It's that animal, the gibbon. Yeah, that little ape. That's the size of a small child. But that includes the animals that are, many of the species that I've discovered are pretty small. They're little bats that you would hold like this, or mice that you could hold in your hand. Others are big, like that big rat. Some of the largest possums, marsupials, arboreal marsupials, tree-living marsupials in New Guinea, turn out to not have their scientific names given to them yet. So there's still big mammals to come, both from New Guinea and other parts of the world that we'll be announcing. One species that you'll see coming out soon from my new posting here in Adelaide will soon be describing the largest species of flying squirrel in the world. And that's something that never got its scientific name. I won't tell you where it's from yet, so we'll wait and see. Yeah.
There's one up here.
Kris, John and Julie Barry from Canalpin. We've been
Hi.
working for many years on paleontology, fossils from the Henschke Quarry at Naracoorte,
Right.
and a very large collection of megalibqualia, Khuskhus material has come from there. We've spent some time with Michael Lee and the people in the museum today looking at a transition where our collection can be put into the hands of caring people. Obviously your passion for echidnas, your name was mentioned, so we said we were coming tonight. That was organised before any of this happened. And with your Mount Anderson Zyglosus browanii specimen, did you find any evidence other than rock art? And do you think there's any chance that you would, after all this time, that it was actually on the Australian mainland?
Yeah, great questions, and thank you for coming from so far, both to make a donation to the museum and to come to the talk tonight. I have to say, I have seen an email with pictures of your echidna fossils just today, and I'm blown away by them. This is the best collection of fossil echidnas that exists anywhere in the world, and is just today, I guess, coming to find its home here in Adelaide, which is wonderful. So that's wonderful, right? Fantastic. So that will form the basis for all kinds of different studies about echidnas and how they've lived here in Australia through time. But the other question about Mount Anderson, so the fact that if this echidna really was living all the way up to 1901, to the point where John Tunney could bring a specimen back to a museum, it offers maybe this kind of small optimism that it might still be out there today. This university has been involved in testing that hypothesis a little bit. Steve Donnellan and other researchers have received scat, like you saw the story of the Pato, scat, echidna dung from parts of the Kimberley to test, because we can test genetically, is it a short-beaked echidna, which we know is there, or is it a long-beaked echidna, which we hope might be there? So far, I guess it's all been short-beaked echidnas. I fear that even if the animal was there in 1901, like the grizzly bear example and others, that it's probably extinct there today. That was a long time ago, and Australia has changed a lot and lost a lot of its mammal species in the interim. So I know that others are mounting expeditions up there. I've heard of a couple of different efforts where people are going to use all kinds of different techniques to see if the echidna is still there. I haven't yet done that, but I will. I'll go look for it. I'll see what I can find.
We'll just go with a couple more. We've got one here.
Kris, David Pickok, we've communicated about quolls across the email. My question is about Australia and hoping to keep your expeditions a bit closer to home. In 2012, we had a Mammal Society meeting in Port Augusta, and there was an interesting presentation by a colleague, Tony Robinson, about the Desert Rag Kangaroo.
Right.
And these sightings where people talk about this little animal being seen at night and disappearing into the dark and lots of things. Do you plan to chase up sort of sightings like that? And do you give them much credibility with this history you have now of finding unusual things? Do you take personal anecdotes by stockmen or things like that with a bit more veracity than maybe the average scientist would?
Yeah, I think that I do, because for several reasons. One is that I've been able to benefit from leads like that to chase up things that are quite unexpected. But the other is that my work constantly is revealing to me, how little we actually know with firmness, about what's living where. Now, the story about the desert rag kangaroo that mentions is a quintessentially South Australian story. It's neat. Back in the early 1800s, a couple of specimens were sent to the museum in London. They realized this is a little desert-living kangaroo that is new to science. It was named and then lost for a very long time. No one saw it again, if I'm getting the history right. But someone, a University of Adelaide student and later technician and academic, Herbert Finlayson, I think his name. Anyways, this is a long story, but he went out on horseback. Quickly, this is great. While a student, he studied chemistry here, not biology. While a student, he was involved in some tragic explosive experiments. He actually lost one of his hands and one of his eyes while a chemistry student here at the University of Adelaide. So I've always thought his greater calling was biology. But he became an incredible biologist. And so the stories in his book, The Red Center, have him on horseback, with other ranchers and Aboriginal compatriots out there, chasing down these desert rag kangaroos and rediscovering them for the first time in 100 years up in the corner of South Australia up near Queensland. So extraordinary stories, and I kind of imagine him on his horse, riding after this fleet-footed marsupial. How is he doing it? And with one hand and one eye, but in any case, he found the desert rat kangaroo, brought them back to Adelaide, and museums all over the world were so interested in the series that he found that they got shipped off to Harvard and London and other places, but we haven't seen it since. And so the question is, marsupials like that, which I think the Aboriginal name is Ulukunta, Finlayson talks about it as a shimmering white ghost kind of moving over the sands, extraordinary. Are they still out there? Is that shimmering white ghost still there? I think some of them probably are. Australia continent is a really big expanse, but cats, foxes, all kinds of things have changed the land and inhabitants. And so, but do I hope to go and look for some of those things? I wouldn't be coming back to Adelaide justice if I didn't do that. Yeah.
Sir, you mentioned extinctions. What do we know? Why do they happen? Is it merely environmental degradation, or is there something else at play that we do not yet fully understand? And the other side, can we hope to see one day the emergence of new species, or is that a one-way street?
Okay, yeah, good question. So why do extinctions happen? For all kinds of different reasons, but usually if there are things that we can see happening on the course of our own lifetimes, it's through some kind of environmental perturbation, right? It's especially in here in Australia, it has to do with mammals, with cats and foxes spreading across this continent. And so, in that case, that's a particular kind of impact. Hunting, like the passenger pigeon or the thylacine. Disease, like the Tasmanian devil. All kinds of different reasons they can happen. But just make no mistake, what we're seeing in the 19th, 20th, 21st centuries are largely these kinds of anthropogenic or human-caused extinctions, something that's very different, game-changingly different from the normal background of extinction that we've always known about, it's by studying paleontology. So extinctions are happening very rapidly, and I think we can all understand that that's going to continue and going to happen rapidly in a human-dominated, fast-changing world. The other question is about new species. Sometimes when I talk about discovering new species or naming species, it can be confusing. What I'm actually talking about in all of these cases are species that have always been there. We just, as scientists, hadn't really found them or realized that yet. So that animal, the Olinguito, has for millions of years been living in those Andean forests. The question whether new species may be born. With mammals, we know from studying genetics in the fossil record, it takes about 1 million years to make a mammal species. It's about how long it takes. So are we going to see that in our lifetimes? No, not really. But that helps us to gauge, the antiquity of all of these different forms of life and what it is that we're losing when each of these things gets snuffed out.
Okay, look, we'll call it to a halt there. Can I thank you, Kris,
You're welcome.
for a start? But please, can I firstly thank all of you. It's a fantastic crowd, and it's a miserable night out there, so I really do thank you for making the effort to come out tonight. And can you just please join me in thanking Kris for giving a great lecture tonight.
Thank you. Thank you.